Welcome back to lesson number 3, DNA, RNA, and protein. What is the difference between these three words? DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. And protein is what you make. So I think you know what the central dogma is. The central dogma says the DNA becomes a RNA, and the RNA becomes a protein. So it's DNA to RNA and RNA to protein. So that's what we call the central dogma of biology. All right, so how is the DNA and RNA involved in the transcription and translation? Among the things you need to understand is what is transcription. Transcription is a conversion of DNA to mRNA. While translation is the conversion of mRNA to protein. So if you know this basic meaning, it's going to really help you to have a better understanding of the rest in the future. So now we want to look at these are the words we're going to talk about in this lesson. Synthesis is a composition or combination of parts to form a whole. Synthesis means you have small bits of something, so then you put them together to produce a whole of something. So that is to say the production of something is what we call synthesis. The geneticists, they accept the basic mechanism for reading and expressing gene is from a DNA to RNA to protein. So this is referred to the central dogma. So the central dogma of biology. So DNA codes for RNA, which guides the synthesis of proteins. We have 3 basic types of RNAs, which you're supposed to know very well. RNA is a nucleic acid, which is very similar to the DNA. But something different between the DNA and RNA is that the sugar ribose in the RNA is not the oxy. It's not losing one oxygen molecule. And it has the RNA has the uracil instead of the thymine. So we have 3 main types of the RNA. Well, messenger RNA, which is represented by mRNA. We have a ribosomal RNA, which is represented by rRNA. We also have a transfer RNA, which is represented by tRNA. We're going to take a look at this and what the functions are. If the function of the mRNA is to carry genetic information from a DNA in the nucleus to direct protein synthesis in the cytoplasm. As you know, there is no ribosomes in the DNA. So after the process of transcription to produce the mRNA actually carries the genetic information from the nucleus to the cytoplasm. The rRNA is associates with proteins to form a ribosome, while the transfer RNA helps to transport amino acid to the ribosome. This will be made clearer to you in the future when we explain the process of translation, so you would have a better understanding of this. So when we talk about translation also, you would have a better understanding of this. Now, what is transcription? Transcription is the first step of the central dogma. So transcription, the first step of the central dogma involves the synthesis of mRNA from a DNA. So it means the production of mRNA from a DNA. That's what we call transcription. The enzyme RNA polymerase helps to regulate the RNA synthesis. The RNA polymerase moves along DNA strand in a 3 to 5 prime direction, synthesizing the mRNA, producing the mRNA. So now this is exactly what we're talking about. The RNA is transcribed in a 5 prime to the 3 prime. So what happens in the beginning is when the DNA, the double-stranded, is being separated, what happens is the RNA polymerase would attach itself to a section of the the single-stranded DNA. So then it's going to have the primer, which is going to be a template for the DNA. So when that happens, so the RNA would the new nucleotide bases will start forming the mRNA. So as you can see, this is one single strand, this is another single strand. So what would happen is this one will be a template to produce mRNA. So let's say, for example, there is A here is going to code for U instead of T. So then it moves forward. Then the C codes for G, it pushes it forward. The U codes for A, it pushes it forward. So you're going to have a 5 prime of a nucleotide strand, I mean the single-stranded mRNA, which they work with the bases like this. So the bases will be formed and it's going to be pushed out. So the main is there is no new DNA produced in this case, but there is a formation of RNA from the DNA single-stranded band. So how is the processes? What are the processes involved in RNA after the production, after it's been made? Because we have a stage before the translation process. So in comparing the DNA code with the DNA code, which it's with its transcribed mRNA, you want to compare the DNA with the mRNA code. The scientists found that mRNA code was much shorter. They found out the RNA is more shorter. So why? The DNA sequence is not found in mRNA are called introns. So the DNA sequences which we don't find in RNA, we call them introns. So introns is a short form of intervening what do you call it, intervening sequences. So they're just standing in between the exons. So the exons are the main ones that carry the DNA or carry the genetic material, but the introns just be in between them just to stop the exons from having contact with each other. So this is the introns. So the introns are just standing in between the exons, and they have to be spliced away. They have to be removed. So before the process of translation will start. So the meaning is in the process of splicing, the introns are being removed and exons are being joined together. Now, the next thing we need to understand is the scientists hypothesized that the instructions from the protein synthesis were encoded in the DNA. So experiments was done in 1960, which actually demonstrated that the DNA code was 3 base codes. So the 3 base codes in the DNA or the mRNA is called the codon. So we're going to have the codes, we're going to explain to you now how the codon is being formed. So here is a dictionary of genetic code. We will notice that all of them, but 3 codons are specific for an amino acid. So we have 1, we have 2, and we have 3. What is the third one? It's another 1, which would code for stop codon. Okay, here. So where you have U A U A G U A G, they both code for stop codon. And when you have U G A, it also codes for stop codon. But all the rest would code for a specific amino acid. So this is how to use a dictionary. So that's the reason when you have a stop codon, the meaning is the process of translation will stop. So now let's talk about translation. After the synthesis of the mRNA, after we produce a mRNA, so the mRNA would move from the nucleus into the cytoplasm. So where it connects at the 5' to the ribosome, so it connects the 5' to the ribosome. So because the 5' prime is set to recognize the ribosomes, so the mRNA code is read and translated into a protein through a process called translation. Translation happens when the tRNA molecule would act as an interpreter of the mRNA code sequence. The tRNA is activated by an enzyme that attaches to specific amino acid at the 3'. So when that happens, the middle of the folded tRNA contains an anticodon, So an anticodon is a complementary sequence to the mRNA. What do I mean by this anticodon? Now let's say we have a tRNA. We have a tRNA which is carrying a codon. Let's say the codon is UAC. So the anticodon for UAC, let's say we have a codon here. We have a codon which is A, and which is U and G. I think AUG is a start codon. So what I'm saying is, let's say we have a codon AUG. So the anticodon for the AUG will be because we said any adenine would have a uracil. There's no thymine. Every uracil will have adenine, and every guanine will have a cytosine. So this is the reason why this is a codon. So this one below is a codon. So it's 3 sequences. This is the codon on the mRNA. They're going to continue to have several nucleotide bases. And this one on the top is anticodon. So the meaning is anticodon is a supplementary basis for the codon. So you're going to go back to your dictionary and see what does a AUG code for. So AUG code for a start codon, which is methionine. So then this is going to be METs. So it's going to be METs. So when this is dropped, another one will come over and it's going to bring its own amino acid and it's going to stick to MET with another, wherever it is. So this is the process. So the middle of the folded tRNA contains an anticodon, a complementary sequence of the mRNA codon. So the mRNA codon, which is this, is having a complementary sequence, which is called the anticodon, which is this. Now let's talk about the role of the ribosome. Ribosomes provide a site for protein synthesis. As we all know, the major role of ribosome is for protein synthesis. So when mRNA leaves the nucleus, the two ribosomal subunits come together. We have a small subunit and we have a large subunit. So they form like this. It is in between these that we're going to have the mRNA would carry the codon through. So they come together and all the mRNA in place for translation. The ribosome structure has grooves that serve as tRNA site for amino acid attachment. So after the whole process, we had a one gene, one enzyme theory, which was proposed by Biddle and Tatum. So in Biddle and Tatum experiment, it showed that one gene code for one enzyme. So we now know that one gene codes for one polypeptide, not enzyme. So how did this experiment, how did it go? So what happened is there was a growth of neurospora on a minimum medium. When you say a minimum medium, it means the medium that has no amino acid. So what happens is the spores were exposed to X-ray to cause mutation to the spores. So then the spores were grown on a complete medium with no amino acid. So, before this is a minimum medium and this is a complete medium. So what happened is after some time, the growth on a complete medium was noticed. There was growth on it. But what happened is on the minimum, there was no growth. So what happened is now the spores were transferred from the minimum medium, and we added supplements to it. So spores transferred to the spores in the minimum medium were added to, supplements were added to it. So what happened is spores transferred to minimum medium are supplemented with the various substances. So we use we use minimum control, we use folic acid, we use thiamine, we use choline, we use pyridoxine, we use arginine. So amongst all of them, it's only arginine that actually triggered the growth of the spores. The other ones did not make the spore to grow. So what does that mean? The meaning is there is one specific gene. The gene, one of the main polypeptide, which is responsible, or the main substance, which is responsible for the generation of the growth of the spore, is arginine. So the other ones does not allow does not trigger the growth. That is to say, one gene codes for what? One polypeptide. So this is all we have for the lesson number 2. So you can attempt the questions yourself, and I'm going to meet you in the next class.